So I know I said I would give you reading to do on your own for the different types of pathways for photosynthesis, but it's kind of hard. And I think what I'd rather do is tell it to you like a story and see if that helps. So you'll have places in here where you'll stop and add a little bit of notes, but it's not going to be a real heavy set of notes. So there are questions on the strips, but we're just going to do this instead. Okay? So, all right, work with me. So this is exactly what we've seen so far. In our minds, this is the way that a section of a leaf of a plant looks like. So we've talked about how that you've got this epidermal layer on the top and the bottom. Both of them are covered by a waxy cuticle to reduce water loss. And at the very bottom of the leaf, you have these guard cells that make up a stomata, and they open, and they're open in order to let oxygen and carbon dioxide gases move in and out of the cell. And the problem is, when they're open, water will also leave the plant. And water management is like the major concern of plants. And, I mean, you know, you've all slept up and had your mouth open all night, and you know what it's like when something's open, how much water loss. In fact, just breathing during the day, you can lose almost a liter of water from your open mouth. So plants struggle with balancing opening and closing the stomata because of water loss and the problems that you get when the stomata is not open and you can have free access to carbon dioxide coming in order to do photosynthesis. So let's remind ourselves about this arrangement of the leaf. So here's the stomata, and then you have this spongy mesophyll or parenchyma, and you've got all these air pockets. And then you, even though these palisade mesophyll cells are pretty tightly packed, there's actually a lot of airspace in here. There's also a type of cell called a bundle sheath cell, and those in a C3, which is like when you cut a leaf from a tree, they don't have any chloroplasts, they're just around the vascular tissue. So the chloroplasts right here in these mesophyll cells would be the place where the photosynthesis occurs. And there's tons of air pockets that expose these cells to carbon dioxide and oxygen. So here's our problem. So here's my story. Remember we said Rubisco is old. Old, been around a long time. And very slow rate of adding carbon dioxide to Ruby P and Calvin. And here's the dilemma. When you've got the leaf set up like we have here, where that you have all of these air pockets that you're allowing the oxygen and the carbon dioxide to come in through the stomata, because the chloroplasts are here, and because both of those gases freely diffuse across membranes, your problem is Rubisco in those chloroplasts has exposure to both carbon dioxide and oxygen. And we said that Rubisco, I mean, we always were making fun of him like he's old and he's slow and he's confused. I don't know whether I'm binding oxygen or whether I'm binding carbon dioxide. Well, it depends. If you have a lot of carbon dioxide, then he'll bind carbon dioxide to Ruby P and go Rubisco. You did a good job. But if the oxygen concentration is high, then he'll change and he'll bind oxygen, which is not going to be productive. So let's think about our dilemma. The real dilemma is, if we're just going to be this kind of silly story and we're personifying Rubisco, he doesn't make good choices. If there's plenty of carbon dioxide, that's what he uses. If there's plenty of oxygen, that's what he uses. So he's not really somebody, since we're saying a story, who should be put in the same space with both carbon dioxide and oxygen, because he'll choose whichever one. this is the process. So we've talked about this before. So rubisco is the enzyme. Enzymes are proteins. Proteins have optimal temperatures that they work at. So rubisco's optimal temperature is lower, cooler. And when there's a lot of carbon dioxide, so we're looking at this blue side, rubisco behaves and will take carbon dioxide and bind it to RuBP, which is ribulose bisphosphate, which is a 5-carbon sugar that's at the very beginning of the Calvin cycle. And we've looked at the Calvin cycle. The Calvin cycle uses ATP and NADPH from the light reactions, and they're going to provide the energy to create a sugar through the Calvin cycle. And what that means is that the energy from the light reactions that you've placed into ATP and NADPH will be transferred over to a storage sugar that the plant can access at any time. So even when photosynthesis is not going on, it'll have storage energy. Okay, if there's oxygen higher than carbon dioxide, rubisco will add oxygen to RuBP instead. This is a process called photorespiration. And the conditions that this occurs in is when the temperatures are higher and that spongy, the air-filled places, there's more oxygen than there is carbon dioxide. Well, this is not a process that makes a storage sugar. It just, we'll look at the diagram here in a minute, but what it does is it consumes ATP and NADPH and gives you nothing. Okay, so it's going to be a process that's wasteful. So right now you need to pause the video. You need to write what is down here in your notes. Let me talk about it for just a second. So if the plant needs to close its stomata, like shut your mouth, you're losing too much water, then you've got a closed system. And what's going to happen is plants are going to continue going through photosynthesis, and you're going to have oxygen released from photosystem 2 where water's split. So your oxygen concentration in those air pockets inside the leaf cell are going to increase in oxygen concentration. And you're going to continue to do carbon dioxide binding to RuBP through photosynthesis, so your oxygen will go up and your carbon dioxide will go down. The very conditions that push the whole rubisco toward photorespiration. So closed stomata change the conditions, and they change the conditions in such a way where that they increase the oxygen concentration, decrease the carbon dioxide concentration, which drives rubisco to do photorespiration. Okay, so copy those notes down. So let's talk about, again, the pathway that we've already learned, which is called the C3 pathway. About 85% of plants go through this. It's the most common way to do photosynthesis. And there's, all trees, the different types of grains that we use for cereals, barley, oats, rice, wheat, alfalfa, cotton. You've got eucalyptus, and you've got sunflowers, soybeans, sugar beets, potatoes, tobacco, peanuts. I don't know why tobacco's on there twice. And then some of the lawn grasses. So very common way of going through photosynthesis. And here's the problem. When the stomata are open, now we're not trying to go into the hole when they're closed and force them to changes, but we're just saying in general, because rubisco is in chloroplasts, in the mesophyll, and the mesophyll tissues have air surrounding them that comes from the gases in the air have free access to the mesophyll through the stomata. Rubisco, unfortunately, is exposed to both oxygen and carbon dioxide. When he is exposed to more carbon dioxide, temperatures are low, he is going to add carbon dioxide to Ruby P. We're going through the Calvin cycle like we're supposed to. He makes a sugar, so the sugar will be made in the Calvin cycle, and you will, basically what you're doing in this particular process is you're capturing the energy that you stored in the bonds of ATP and NADPH from light. So the light reaction's job is to take light energy and put it into carriers, ATP and NADPH, and hold that light energy and take it to the Calvin cycle so that that energy can be used to make bonds where you store that energy for later use in sugars. So in this particular case, it's about 37% efficient. So if you consider that the light energy you were able to trap in ATP and NADPH as 100%, Calvin will use those molecules, ATP and NADPH, and be able to take out of that 100%, 37%, and you save that in the storage molecules of sugar in the G3P. You lose 63% to heat, and that's just going to dissipate. It can't be used for work. The 37% efficiency is really not bad for living things. So this is the Calvin cycle. This is what Rubisco is supposed to do. Way to go, Rubisco. So this is just a diagram. You may want to stop the video and put this in to your notes. But it's showing that in these mesophyll cells, there's access to these gases that come in through the stomata at the bottom of the leaf. Carbon dioxide, oxygen, but in this case we're going to focus on carbon dioxide. It's nonpolar. It'll come straight in through the membrane. Rubisco uses it. It will fix it in the carbon fixation stage and attach it to Ruby P, and then you'll end up with a sugar when you're done where that you've used the energy in the bonds of ATP and NADPH and you've saved it in the bonds of the sugar. So this is the C3 pathway. Unfortunately, because in the C3 pathway Rubisco also is exposed to oxygen, you can have photorespiration. So let's look over here on the right first, because this is what we've learned. When carbon dioxide is in high concentration, temperatures are low, Rubisco, which is an enzyme, will add carbon dioxide to Ruby P, and you'll go through the Calvin cycle. You'll use ATP and NADPH, those molecules from the light reactions, and that energy that you brought in those molecules will be moved into sugar at around a 37% efficiency rate. On the other hand, if Rubisco instead is exposed to high concentrations of oxygen, which happens often when the stomata are closed, then Rubisco will instead add oxygen to Ruby P. You do not make a sugar. You do use ATP and NADPH, but you don't get anything for it. You release carbon dioxide, you get no sugar. You made no storage molecule. So you wasted what you had been provided, and you don't have anything saved. Okay, so how can we help Rubisco with this problem? So this process is called photorespiration because Rubisco, in this case, will bind oxygen to Ruby P. So we're consuming oxygen, and when we're done, we don't have a sugar, we release carbon dioxide, which we're supposed to be putting into a sugar, like we're sending it back out into the atmosphere. So because cellular respiration also consumes oxygen and releases carbon dioxide, that's why this is called photorespiration. They're different processes. They're just making the point that this is not photosynthesis. It is very energy and carbon wasteful. So is there some way we can reduce the effects of this photorespiration problem, which happens because in C3 plants, rubisco is exposed to oxygen? Well, there's another pathway called the C4 pathway. Sugarcane, I don't know why millet's on here twice, tall grasses, corn, those are probably the most common ones that go through C4 pathway. So what does that look like? Well, the leaf is arranged differently. So in this way, you do have the same epidermal layers, stomata at the bottom, they're open, but you've not got the spongy mesophyll. You don't have places where that air just moves all throughout that tissue in the leaf. So here's the other thing that's different. Here's your mesophyll cells, and here's your bundle sheath cells, but the bundle sheath cells in a C4 plant have chloroplasts. So if we go back, to here, you'll notice not only are there air pockets, which the C4 plant doesn't have, but the bundle sheath cells that surround the vascular tissue don't have chloroplasts, so could not do any portion at all of photosynthesis. But in the C4 leaves, you're going to see that these bundle sheath cells have chloroplasts. So we have this ability to, I'm going to come back to that one, to separate the process of the Calvin cycle and move it to the bundle sheath cells and only let the mesophyll cells be exposed to the gases that come in through the stomata. So if we look here, this is a really good way to see the difference. See all these air pockets in the C3 plant? So you have this carbon dioxide, you have oxygen come in, and it's going to be freely distributed within all of the tissue of this mesophyll. And so you're going to do photosynthesis in any of the cells that have chloroplasts, but all of the cells that have chloroplasts are those parenchyma cells, and they're all exposed to both carbon dioxide and oxygen. You don't have any way to save rubisco from himself, like to separate him from the gases. This anatomy, this structure of the C4 plant makes things much simpler. So instead you still have epidermal layers, cuticle, stomata, but the only cells that are exposed to the gases that come in and out through the stomata are these parenchyma cells right here in the mesophyll. Then you have inside, away from, separated from the gases, these bundle sheath cells, which you have right here as well. But look at the difference. In a C4 plant, they have chloroplasts. So they're like, hey, give me Calvin cycle with rubisco. Let me help separate rubisco from the gases. So for our story, this would be rubisco. We can put his process over into the bundle sheath cells, which they don't have exposure to the oxygen or the carbon dioxide. So these would be the mesophyll cells that are the parenchyma. They have chloroplasts. They can go through carbon fixation. Rubisco also is in a chloroplast in the bundle sheath cell, but we've separated the two. The problem is, and the thought is, how are you going to fix the carbon in this mesophyll cell if you don't have rubisco? Well, it's with a different enzyme called PEP, P-E-P, carboxylase. And he's not got the problem of, well, I don't know, going to bind oxygen. I'm going to bind carbon dioxide. He is like, hey, I'm on it. We're just doing carbon dioxide here, people. I always call that monogamous because that's what enzymes should be. There should be one substrate. So this PEP carboxylase will not be influenced by the concentrations of oxygen. It is not a substrate. It will only bind carbon dioxide. So it binds carbon dioxide, and then that carbon dioxide is what is passed to rubisco in the bundle sheath cells. So rubisco, hey, guess what? You don't get to make a choice anymore. Yeah, you could bind both oxygen or carbon dioxide, but we're not giving you any oxygen. We're just going to give you carbon dioxide. Go with that. So this is what it looks like, and you may want to pause the video and draw this, but you've got those mesophyll cells that surround the bundle sheath cells. They're the cells that have that carbon dioxide and oxygen that can freely move into and out of their cell membrane. So carbon dioxide comes in, PEP carboxylase is like, I got it. I will bind you to PEP. We'll make a 4-carbon molecule so we can save you carbon dioxide. That's where the C4 comes from, because we make a 4-carbon molecule first, and then that 4-carbon molecule is transported to where rubisco is, which is in the bundle sheath cells. They're in that middle part that they're not exposed to the gases of carbon dioxide and oxygen from the stomata being open or closed. And rubisco only will get carbon dioxide because there's an enzyme that's going to break carbon dioxide off of the malate. It creates pyruvate. There's another enzyme that takes pyruvate and takes it back to PEP. It will cost you an ATP to do this step, but you won't do photorespiration. You can't, because rubisco will never get the oxygen, just the carbon dioxide. So he's going to do the Calvin cycle, and he will always go through the Calvin cycle correctly, where the carbon dioxide is bound to RuBP by rubisco, and when you turn, you end up with a sugar where you're saving some of that light energy. So the C4 pathway goes and uses location as the way to reduce photorespiration. In other words, help rubisco not make stupid choices. So there's another alternative way to do photosynthesis, and it's called the CAM pathway. So this is a really weird word. It's crassolution, and so basically what these are succulents. So they're like cactus, pineapple, Spanish moss. There's different orchids, but they're going to be, most of them you're going to think of as succulents. So here's the thing with them. They look a lot like a C3 plant, except they're, you've seen them. Like I'm over here looking at my little cactus plants I have in my penguin and my sloth, and I don't know what that other one is. But their leaves are thicker, and that's because their cells are larger, and they do not have big air pockets. They don't have that spongy. If you pulled that leaf apart, it wouldn't be like pulling the leaves apart where you get that kind of spider webby looking stuff, which is where your spongy stuff is. So there's not this separation where bundle sheath cells have chloroplasts. They don't. They're just like the C3 cells. So how in the world? If C3's got a problem, won't the CAM have a problem? Nope. The CAM, these plants thrive in high temperatures. So that's a huge problem anyway, because that's how you start the photorespiration part, is that I have a high oxygen concentration and temperatures are high. What it really means for the CAM plants, let me move my head right here, is that they cannot keep their stomata open during the day. That's a huge big deal. You've got to have carbon dioxide coming in for photosynthesis to occur, because photosynthesis occurs during the day where there's light, because the light reactions are going to make the ATP and the NADPH, and then Calvin uses those products, and Calvin might be able to run a little while without the light, but eventually Calvin's going to run out of ATP and NADPH. You have to have the ability to have carbon dioxide during the day for photosynthesis. And as soon as you close the stomata, you're not bringing any in. So what CAM plants do is that they, come on, work with me, what's your deal? They change their work in photosynthesis based on time. So once the light's gone and the light reactions, the photosystems aren't making any more ATP or NADPH, Calvin's going to be like, hey, what happened? Wait, I'm like, I could work, but where's my stuff? So if there's no ATP and no NADPH, because there's no light for the photosystems, Calvin stops working. And so in the dark, Calvin stops working. So this is Rubisco taking a nap. He's inactive when there's no sun. And CAM plants also have PEP carboxylase, but they don't have to separate them into two separate places because they separate them into two separate times. So at night, when the stomata are open, because the temperatures are cool and there will not be this horrible loss of water like you'd have with open stomata in a hot desert in the day, then you're going to bring in your gases through the open stomata. PEP carboxylase is like, I'm binding to the carbon dioxide. Does the exact same process where that carbon dioxide is fixed. So let's look at that. You have carbon dioxide, oxygen too, but at night, PEP carboxylase will work the night shift, add the carbon dioxide, fix it. It's also called sequestering because you're putting it off like by itself in a way to hold it. They use that word for juries. Like if somebody's ever been on a jury and they've been sequestered, it means they've been kept away from everything else and put off someplace. So the carbon dioxide comes in, PEP carboxylase at night, works the night shift, attaches it to PEP, so fixes it into a 4-carbon sugar, or a 4-carbon, actually it's not a sugar, it's an organic acid. So puts it into malate, and then we wait till it's daylight. So when it's daylight, you've got malate, which is malic acid, same thing, will be transported to and around an enzyme where that it will break off that carbon dioxide. Now we're not in different spaces, we're still in one mesophyll cell, but at the different time, the enzyme will come on during the day, take that carbon dioxide back away from malate, and it doesn't show it here, but it'll also make pyruvate, and that pyruvate will then be changed into PEP, and it will cost an ATP. So this also costs, but you're only going to let Rubisco have carbon dioxide. So you have no photorespiration going on. So the difference in the CAM pathway is because these plants live in very dry, hot places, and their stomata cannot be open during the day. They have to be closed during the day and open at night. And so you run this carbon fixation cycle while the stomata are open at night, so that way when you close them during the day, and Rubisco's like, ready to go, I'm up, I'm ready to go. Where's everything? Well, the light reactions will be sending ATP and NADPH, and then he has to have carbon. That's what his job is carbon fixation. And they've provided that through the night shift, where they took the carbon and then held it in malate. So then it's released in the daytime over to Rubisco to bind with RuBP. So pause the video and let me walk you through this. The whole point is we're helping Rubisco not make bad choices. Bad choices using oxygen and going through photorespiration. So what we've done is we've got alternative photosynthetic pathways. The C4 plants are going to separate the processes of carbon fixation and the actual reduction, which is the making of the 3-carbon sugar, into 2 separate cells. And that way Rubisco is in the cell that will not be exposed to the oxygen. So he can't be like, well, I don't know what I'm doing. It's like, yes, you do. All you have to do is one job. There's the CO2, bind that. So this is about separation by space. The CAM plants all live in the same little room. Rubisco's not sent off by himself, but when he's not active, when he's sleeping at night, they do the carbon fixation through that PEP carboxylase cycle, and they pull the carbon dioxide in while the stomata are open at night, hold the carbon, and then during the day, when the light reactions are working, and Rubisco through the Calvin cycle has everything he needs, they're feeding carbon dioxide from that carbon fixation that happened at night. So put those notes in there. And then I did want to just remind you exactly what photorespiration is. It's the binding of oxygen. That's why it's called photorespiration. It consumes oxygen. So Rubisco consumes oxygen because it's bound to RuBP. And then you use your light reaction products, ATP, NADPH. Calvin always uses those, but you don't get a sugar from it. You end up releasing a carbon. Your job was to take carbon and bind it, not release it. So the reason why it's called photorespiration is because it consumes oxygen and releases carbon dioxide, like cellular respiration, but you don't get anything from it. You don't get ATP like cellular respiration does, and you don't get a sugar like that photosynthesis does. So here's the last thing I want to say. It's been around pretty much since the dawn of time. We want to be careful as humans, especially when we write responses to the questions, like for the free response questions. You don't want to say things like it's useless, or you can say wasteful as long as you qualify what you mean. But you don't want to be this, put your human values into it. This particular process of photorespiration is part of what Rubisco does. It just is. And Rubisco is old, highly conserved, been around for a long time, exactly like it currently is. It's in 85% of plants, the most common protein in the world. So there must be something we don't know. Processes that are wasteful and useless over time become outcompeted by other processes that aren't. So maybe we're missing something. There must be a reason for this to be conserved. That's all I'm saying. Okay? So I will stop this, put it in Edpuzzle, give you some questions, and I think there's a question about this on the test. That's why I wanted to be sure that I didn't just leave you on your own. So I think you're wonderful. Thanks for hanging in there with me. I kind of like my little Rubisco dude. He's pretty cute. So I think you're great.